morning. Thank you very much for your kind introduction. It's a great pleasure for me having the opportunity to present you today some data from our lab. I'm working at the MLL Munich Leukemia laboratory as a PhD student in the Department of Area Next Generation Sequencing. And we have mainly three different methods: gene expression, SNP arrays, and next generation sequencing. The 454 has different applications: DNA-protein interaction maps, epigenetics, metagenomics, transcriptome sequencing, whole genome de novo sequencing, and in May 2009, when we have started with the 454, we focused on target shotgun sequencing and Amplicon sequencing. Here you can see our next generation sequencing room at the moment at the MLL. We have two GS Flex sequencer and two GS Junior. What do you can see on this picture? It seems to be like honeycombs with colored circles, but instead of that, it is a detailed view of such a picotitter plate where on the surface are 3.7 million wells. But now from the beginning, when we have started to establish the 454, we validated it with two different projects, with the Amplicon project and the Nimble Shotgun project. First of all, I would like to start with the Amplicon project. We selected therefore 96 patients. 81 were harboring a chronic myelomonocytic leukemia, and other were malignant cases with known mutations. We sequenced or screened these patients for 7 candidate genes: TET2, RANKS1, JAK2, SIBL, MPL, NRAS, and KRAS, and had therefore 43 different amplicons. The chronic myelomonocytic leukemia is a clonal hematopoietic malignancy characterized by features of both a myeloproliferative neoplasm and a myelodysplastic syndrome. And we focused on this cohort for mainly two reasons. First of all, because it was described very less on a molecular level, and secondly, because in our cohort, more than 80% showed a normal karyotype. Here you can see our 7 candidate genes. For the first 5 genes, we sequenced only the known hotspot regions, whereas for the second 2 genes, we sequenced the complete coding region. Here you can see our non-454 workflow. First of all, the sample entries. Then we take the blast from the Foucault gradient, and we can isolate 12 samples per run, isolate the DNA using the QIAcube from QIAGEN. Then we generated the different amplicons for every patient. In total, we generated more than 3,500 single PCRs. Then we purified the DNA amplicons, we quantitated the DNA, and then we pooled them in an equimolar way, meaning that we had for every patient 1 single tube, and this was a mix of 43 different amplicons. Then we started with the 454 workflow. Here you can see the Emulsion Pcr. We massively we clonally amplified the DNA using the Emulsion Pcr. This is a water droplet in oil mixture, and in the best case, we have 1 DNA bead and 1 DNA molecule in 1 water droplet. Then we had to broke the emulsion and to enrich the DNA beads with the DNA molecules. Then we massively parallel sequenced using the 8-lane picotitter plate. In our first study, we didn't use MIDs, therefore we sequenced 8 patients per plate, and in 12 runs we generated the data from all patients. Here you can see the inner life of such a platform. We had to load our picotitter plate with different layers, with DNA library beads, enzyme beads, and backing beads. Here you can see the detailed view of one of the well on the surface of the picotitter plate, and the big one is the DNA bead, and there are also enzyme beads which contain the sulfurylase and luciferase enzymes, and the backing beads which retain the DNA bead into the well. So now we know that these are not honeycombs, but successfully sequenced beads. Now we can speak about reads, and the red ones are failed and the green ones are successfully sequenced. Here you can see the flow of incorporated nucleotides. It seems to be like the Sanger sequence, and there is incorporated, is there a laser pointer? Oh. It's incorporated, you can see here for example 1G, 1C, 1A, 1G, and so on. On the scale, you can see how many nucleotides are incorporated. Here you can see our first results. We generated up to 500 base pairs long reads, and in median we get 840-fold coverage. The coverage is the same word as generated reads, and we need a very high coverage for good sensitivity. So for example, with 500-fold coverage, we have a sensitivity of about 5%. This is a detailed view of the Amplicon variant analyzer software. For example, for 9 patients, patient 13 has a mutation in Sybil exon 8 and Sybil exon 9. And for example, patient 21 has a mutation also in Sybil exon 8 and in JAK2. You can see here a lot of mutations with a mutation load over 20%, but also a lot of mutations under 20%, which is under the Sanger sequencing burden. We validated also these mutations. For example, here we detected a mutation in the common codon 617 in JAK2. We generated here 606 reads and detected these mutations with a mutation load of 1.16%. We validated this mutation using the melting curve analysis and detected the mutation with approximately 1%. So here you can see our results detected in the 81 CMML patients. TET2 was the most mutated gene with 34.4%, then Sybil and NRAS with 22%, KRAS with 12%, JAK2 with 10%, RANKS1 with 9%, and we didn't detect any mutation in MPL. So in summary, we detect an overall mutation rate of 72.8%. JAK2 was first described at the ASH meeting in 2008. The specific function is not yet known, but it has epigenetic function. And the mutations are predominantly localized in the conserved regions, but spread over the whole exon, over the whole gene. This analysis we performed in 2009. In 2010, there were other very important and interesting genes published in the literature, such EZH2. EZH2 encodes the catalytic subunit of the polycomb repressive complex 2. This is a highly conserved histone 3 lysine 27 methyltransferase that influences the stem cell renewal. EZH2 is the methyltransferase, and UTX is the antagonist of and is a demethylase. Here you can see also TET2 and JAK2 on this schematic picture. And EZH2 function as an oncogene or a tumor suppressor gene in different diseases. So in 2010, we looked again in this cohort for IDH1, IDH2, NPM1, ASXL1, and EZH2, and we detected an overall mutation rate of 81.5%, and we detected 9 mutations, 9 patients mutated for EZH2. Here you can see the survival analysis about EZH2. EZH2 has the most important impact or survival impact analyzed thus far. EZH2 mutated patients in comparison to EZH2 wild-type patients after 3 years. EZH2 mutated patients are alive, 40% are alive versus nearly 70% of EZH2 wild-type cases. In 2009, we detected also that the 2 mutated patients show a better survival than the 2 wild-type patients, and so we subdivided our cohort into 3 distinct groups: the 2 mutated, EZH2, the 2 wild-type patients, and EZH2 mutated patients. And after 3 years survival, 33.9% of EZH2 mutated patients were alive, nearly 50% of EZH2 and the 2 wild-type patients were alive, and nearly 90% of the 2 mutated cases were alive. This is another very interesting aspect of the 454. Here, a patient is harboring 3 different mutations in KRAS exon 3. There are 3 different mutations in the same amplicon in codon 12, codon 19, and codon 20. And you can see here that the mutations never occur in the same read. So we can subdivide here between a monoallelic or biallelic or different subclones. So in my first summary, we can say that the number of molecular markers used to categorize myeloid neoplasm is constantly increasing, and NGS screening allows a comprehensive characterization of the molecular background in CMML. In summary, we detected 81.5% of CMML cases mutated, and NGS outperformed conventional single marker mutation screening and additional revealed novel molecular insights in this entity. Now we have the 454 for 2 years, and we are focusing on 4 different aspects: in disease characterization and classification, in prognostic information, predictive information, and also in research. And you know that we are a diagnostics routine laboratory, so we spend a lot of resources and a lot of time in automation solution, for example, for PCR setup, PCR purification, and enrichment. First of all, a few words for PCR setup. We designed together with Roche assay-on-demand plates for the 2 Sybil, KRAS, RANKS1, and RANKS1. And the 2 is quite a big gene. We subdivided it into 27 amplicons with a median read base pair length of 343. In combination with Sybil and KRAS, we are able to amplify the three different patients using the 96 well plate with lyophilized primers from Roche Applied Science. But my favorite enrichment method is the Fluidigm 44.44 axis array, because it allows to amplify 48 samples across 48 primers in one single PCR step. So after one step, I have generated more than 2,300 distinct PCRs, and the hands-on time is about 1.5 hours. For the purification of PCR products and the preparation of quantification, we used the Beckman NXP from Beckman Coulter. And we use it also in combination with the RAM system for the enrichment of the beads. And what's about data analysis? We are using the Amplicon variant analyzer software from 454, and the University of München has written an HTML report that allows us to visualize the quality report, so to check the forward and reverse coverage and the sequenced reads. But our favorite analyzer software, or much used, is the GSI medical system. Here you can see, for example, a 7 base pair deletion in exon 4 in RANKS1, a mutated patient, and it has a lot of advantages in comparison to the AVA software. For example, you can see here immediately if it is a SNP or a mutation, and also the amino acid change. Now I would like to talk about the IRON study. The IRON study is the interlaboratory robustness of NGS, and there were different participants. We from Munich, then Professor Basso from Padova, Dr. Simon from 454 itself, Professor Vandenberge from Belgium, Dr. Garchichochea from Brazil, Dr. Janssen from Nijmegen, Professor Martinelli from Bologna, Dr. Gabriel from Linz, Professor Young from London, and Dr. Timmermann from Berlin. We selected for this study 18 samples from our Amplicon study, for our first study, and shipped them out in this blinded sample, shipped them out in combination with 454 reagents, enzymes, 96 well primer plates, and the study protocol to the different centers. Here you can see our patients. The patients were harboring mutations in TET2 and/or Sybil and/or KRAS. Here you can see first results. In here you can see that the median coverage was 689, and the coverage was very homogeneous between the single amplicons. But it was also very homogeneous when we have compared the different centers. Here, for example, the center 5 in comparison to the other 9 centers. But this, I think, is the most interesting result. Here you can see homozygous and heterozygous mutation, and even the coverage is very different, very variable between the different centers. Here, for example, we have a more than 1,000-fold coverage, here less than 200, but the mutation load is nearly always the same. So the summary of this study is that an international consortium demonstrates the robustness, precision, and reproducibility of 454 Amplicon next generation sequencing technology across 10 laboratories from 8 countries. And we had a lot of experiences as a sequencing center, but there were other countries or other laboratories which they has already started with sequencing. Secondly, the observed variability was uniform throughout the range of the measurements, underlying the high consistency of the variant results between any 2 laboratories. And now, more detailed analysis are ongoing of mutations with frequency less than 10%, and we has already started with the iron study phase 2, where we are focusing on disease-specific working groups, for example, for DALL, MDS, or CML. Now I am arrived to the last part of my talk, to the targeted shotgun sequencing. Therefore, we selected 48 samples for a 2-lane pico-theta plate. We incorporated here also MIDs and used the titanium chemistry. In our first study, we didn't use the titanium chemistry because there was, again, the old chemistry. There are different sample preparation strategies: PCR amplification, microarray sequence capture, and in-solution sequence capture, and we decided as for microarray sequence capture from Emulsion. We designed a chip with 95 different candidate genes. For 92, we sequenced only the exons, and for MLL, ranks 1, and CBF beta, the full coding region. The exons and introns, and so we had a total capture of 1.91 megabases, and 96% of bases were covered. The 454 workflow was very similar to the Amplicon strategy, but the library preparation was totally different. We used the old protocols, so 20 microgram of genomic DNA were needed. The hybridization was 72 hours, and the median enrichment in our cases was 294-fold. Here you can see nice results. We detected insertions, for example, 63 base pair insertion in FLT3 ITD, and the 3 base pair deletion in KIT codon 419. But these are the interesting results. We were able to detect chimeric reads and therefore detect balanced aberrations. This is such a chimeric read. The first part mapped to chromosome 16q, and the second part to chromosome 16p, and so we detected this fusion CBF beta and MYH11. We didn't detect only known mutations and known fusions, but we were also able to detect unbalanced aberrations and novel fusion partners for ranks 1 and MLL. So, thank you very much for your attention.
Thank you very much, Vera. So the presentation is open to discussion. So if you have some question, please. Vera, I have a question. So do you think that next generation sequencing is ready to go in diagnostics, at least in hematology?
Yes, we are sure for several genes we use only next generation sequencing, and it's interesting for disease classification, but overall for disease monitoring. because we have a higher sensitivity than Sanger.
Okay, thank you.